So we're going to move on to the next talk, and the next speaker we have is Dr. Michael Yassa. Michael Yassa is, in fact, also an assistant professor at UC Irvine. He is a member not only of UCI MIND, but also the Center for Learning and Memory. Now, Dr. Yassa has started his lab originally at Johns Hopkins University as an assistant professor, and then decided to come join us again at UC Irvine. So one of his laboratory is really focusing on neuroimaging and looking to see if there are some mechanisms of memory that are involved early on and then are more defective in the aging individual. And so he's got a very impressive publication record as well. And in addition, he really does like to talk about his work. And he's given over 100 public lectures, both to scientific and lay audiences. And so he's going to be telling you his current work on looking at neuroimaging and the tools that they're using that might be able to lead to better clinical evaluation of better clinical trials in the future. Mike.
Okay, thank you all for being here, and hopefully you'll allow me the honor of introducing you a little bit today to some of the imaging research that we've been doing over the past few years. And I think the real goal from this talk is to really get across the complexity of neuroimaging data, the multiple different modalities that we can acquire imaging data in. And even though typically imaging is thought of as something that you do as a diagnostic, you go get an MRI on your knee or on your head maybe to diagnose a stroke or something like that, there's a lot of research uses that we want to talk about as well that can allow us to develop and design better clinical trials. So that's what I'll try to focus on today. And I like to start with my take-home messages just to make sure that everybody's on the same page. And the first thing that I'll talk about is actually a behavioral phenomenon, not so much imaging, which is that memory, obviously, as we all know, does change as you get older, not even in the context of Alzheimer's disease, but even just in the context of healthy, normal aging. There are subtle changes to your memory system that happen. And some of those are very interesting to us, and we've developed some ways with imaging to be able to look at them a little bit closer. We typically use what we call high-resolution imaging techniques, and I'll go over what that means. These are not your standard run-of-the-mill neuroimaging techniques. These are at a fair degree of sophistication and precision, and really allow us to look at memory changes in the aging brain in unprecedented detail, in my opinion. Identifying these changes and the neurobiological substrates of these changes is, in my opinion, critical for the understanding of Alzheimer's disease, and I'll try to make that case very strongly today. And I'll also make a pitch for brain imaging biomarkers. And what we mean by a biomarker is something that can be an indicator for a disease process, something that is changing in your brain or in your body. As you've heard today, for example, there are blood-based biomarkers that one can look at. But the important critical detail here is that this is in the absence of clinical symptoms. It's in the absence of actually having cognitive deficits that are sufficient to diagnose somebody with a disease. So these biomarkers can be very important if we want to look into the preclinical stage of a disease, which is very important for early detection and targeting people early on. And then, if we have time, I'll talk with you a little bit about what I think is really a necessary paradigm shift in biomarker research to start focusing on some of these multimodal brain imaging techniques. And most of the techniques that I'll talk about today are completely non-invasive. They all involve MRI. So I'm actually going to talk about imaging-based biomarkers without talking about amyloid imaging at all. I'm not sure that's entirely possible, but we'll see how far we can get. But the idea here is that focusing on these multimodal brain imaging techniques can actually allow us to say something new and maybe even pick up on new targets that can be looked at in Alzheimer's disease. So let me start by telling you about a phenomenon that happens. I'll talk about rats first, and then I'll tell you how it happens in humans, which is that memory changes, but it's in a sort of a subtle way. And I'm going to refer to this as discrimination memory, being able to distinguish among similar experiences that you might have. And you may not be surprised to hear this after hearing the work from Kim, is that there are a lot of commonalities between rodents and humans. We can certainly test their memory in some of the same ways, although there are some parallels there and some differences. But this is my version of the aged rat there. And this is actually a very simple test. It's not a test at all All you have to do is put the animal inside an environment and let them just walk around. But the critical thing is that while this is happening, there are neurophysiological recordings that are taking place from this animal directly in their hippocampus, a particular region of the brain that seems to be very susceptible to Alzheimer's pathology early on. And what you find is very interesting. So if you just look at a young animal and you're recording from hippocampal cells while the animal is traversing these environments, which I'll direct your attention to just some of the similarities between those environments. They have some of the same cues, but they're slightly different geometry. So one is circular, one is square. And you would think that the animal would be pretty good at distinguishing the fact that these environments are different. So if you put the animal here first and then move them into this environment, the animal should have some recognition that this is a different environment. And in fact, the cells in the hippocampus can do that discrimination. So this cell doesn't really fire in this environment, but it fires heavily in this location, in this environment. You put the animal back in the old environment and that cell stops firing. That cell can discriminate between those two environments very well. But if you do the same kind of recording in an aged animal, you see something very different. If you record from a similar cell in that aged animal, what you find is that that cell can't really distinguish between the two environments. It fires in the same location, fires to the same extent. It says, it's all the same to me. I can't really discriminate that these are two different environments. Now, this reduction in discrimination is very important because we think we might actually have pinpointed the mechanism for why that happens. But let me first talk to you about memory loss in humans so that we can build this picture together. The kinds of things that people report as they get older are things that you guys are very, very familiar with, and you've heard these complaints. Episodic memory tends to go down. Source memory, being able to pinpoint when and where things happened, that sometimes goes down. This ability to have a recollective experience, to be able to piece things back together from prior experiences that you've had, that also goes down. Contextual memory is another variant on this. Spatial memory, certainly getting lost around new spaces and having trouble navigating and so on. And also an increase in false memories is something that tends to happen quite a bit as you get older. In fact, one of the really interesting things that happen as you get older is that your degree to which you produce false memories goes up, but your degree of confidence in your memory also goes up, which is interesting. So you're really, really sure that some things happen to you that are just completely false, that did not happen. So if you're writing autobiographies, that's going to be a problem. But this all shares something in common. There is a massive susceptibility to interference, okay? And the problem here is that there is reduced discrimination in the system. Your ability to discriminate among similar memories, similar experiences that happen to you, goes down as a function of age. Now you might think, that seems like a very peculiar kind of memory deficit, but in fact, it explains a lot of these problems because this is a memory deficit that spans across all of these issues. Think about it. All of the things that happen to you on a daily basis, there's a lot of repetition, okay? You wake up in the same bed every day, you go to the same job, you eat the same food, you talk to the same people, work with the same people, do a lot of the same things, come home to the same family most of the time. And a lot of these things need to be saved essentially as separate experiences. And if there are any problems with that system, any problems with discrimination, you're going to see it in these memory problems. But there's a way to test this more directly. We can develop a task that actually puts a load on your discrimination ability and see how well you do as an older adult compared to young adults. And that's what we've done. So we've taken a task that is very, very simple. All we do during the study phase is we have you look at pictures of everyday objects. And we ask you to tell us if it's an indoor or an outdoor item. Very, very simple incidental type question. We just want you to pay attention to these items. But then later during study, we give you, later during test, we give you 3 choices for every item that you see during test. Some of these items could be exact replicas of what you've seen before, in which case you're supposed to say, I've seen that before, it's the same thing. Some things could be completely new, in which case we expect you to say, that's new, I haven't seen that before. But sometimes we can be really mean, and we can show you things that are very similar but not identical to what you've seen before, like these picnic baskets, for example. Now, in that situation, the question is, what are you going to say? Are you going to tell us it's similar but not identical, or are you going to fall for it and say, that's the same thing I've seen before? And that gives us a pretty good index of how much discrimination has gone down. So let's walk through the data. If you compare older adults and young adults, what you see is that their ability to say to a completely new item, that this is a new item, is unchanged as a function of age. So novel and novel, pretty good performance. If it's a repetition, you're pretty good at saying, I've seen that before. So repetition, target detection, unchanged. The only time you see a difference is when you look at these similar lure items, the tricky items. If you're a young adult, you're much more likely to call that similar, which is the correct response. But as you get older, your tendency to call it old starts to go up. Now, there's an interesting feature about this task, which is that if we didn't have these similar lures, if we didn't have this set of bars right here, if you put those two graphs, close them up together, you find that there's no age-related memory deficit. And that's in fact what most of the prior research has reported with these kinds of object recognition tasks. There is no age-related deficit. It's only when you have these tricky items that you can actually pick up on that subtle kind of change. Now, why do we care about this so much? Well, we care about it because we think we have a mechanism neurobiologically for it. So let me tell you how we look at this with imaging. Functional MRI is a method that kind of capitalizes on two different phenomena that are very, very good to sort of bring together. One is that in your brain, wherever you have an increase in brain activity, local increase in brain activity, some region is working really hard at something, you're going to get a local increase in cerebrovascular flow. So the blood supply is going to go to that region to be able to feed it and provide it with oxygen and so on. At the same time, there's a really interesting phenomenon in MR physics, in magnetic resonance physics, which is that oxygenated and deoxygenated hemoglobin actually have different magnetic properties, and the contrast of those two can be measured. And that's the kind of thing that we're able to measure in an MRI scan. So in an MRI scan, images usually will have a higher intensity for areas that are working a little bit harder. Now, keeping this in mind, how can we use this technique to look at discrimination? How can we use it to look at memory? There's another really interesting phenomenon that happens, and we know about this, we've known about it for quite a while in imaging studies, which is that the first time I show you any new picture, there's lots and lots of activity in your brain. Your brain likes new stuff, particularly the hippocampus, this part of the brain that we're very interested in. So let's say I show you this picture. It's the very first time you see this picture, and your brain essentially lights up and says, ooh, that's new, that's kind of cool. Then I show you another new picture, and your brain does the same thing again, says, ooh, that's new, cool. And now I show you the same old picture again. Activity level actually comes down. Okay? There's a repetition suppression that happens in functional imaging data. Typically, this is sort of an adaptation phenomenon. It says, this is no longer interesting, I've seen it before. So blah, I don't care. But now the critical question is, what if I show you something like this? Which, if you've been paying attention, it's not exactly the same as the one I've shown you before. It's actually subtly different. And the question is, what's your brain going to say? Does it say, is it the same picture that you've seen before and bring its activity level down? Or does it treat it like a new item and keep activity levels up? So it actually allows us to pit against each other these predictions, right? So if you have a first presentation and a repeated presentation, and we know that there's a difference here. First, there's lots of activity. With repetition, there's low activity. Now we can ask the question on these lures, is there low levels of activity or high levels of activity? And if you are discriminating, you should have high levels of activity. This is a process that we've referred to as pattern separation. Many people have referred to it in the same way. This is not unique to us. And if you don't, you're just sort of generalizing across different instances, then you're doing this sort of pattern completion. And this was kind of an interesting first foray into using imaging data to look at these computational principles. And what has been discovered is that the dentate and CA3 regions in the hippocampus, particular regions in subfields in the hippocampus, are very, very good at discriminating. So they're showing a profile that is very consistent with this discrimination profile. So now we've pinpointed a location, a place where this mechanism might happen. And the question is, what actually happens to it as you get older? And the answer is, it stops discriminating as well. And here's how we can look at it. We can actually vary the extent to which these stimuli are similar to each other. So some may be very, very similar, and some may be not so similar. We can vary that across a continuum. And then we can ask the question, at what point during this continuum does the brain start to reflect a discrimination signal? Can it do it very early, even with the most subtle kind of change, or do you need things to be very different in order to say those are different? So those are the kinds of predictions that we had. We thought that the young brains would be able to discriminate very quickly. Okay, so you get a dentate CA3 signal that rises up very quickly, even with the most minor kind of distortion, whereas the aged brain will be much more rigid to that stage. So what you can think about here is that this is sort of the repetition brain, and this is the discrimination brain. Okay? And the data map on exactly to those predictions. So what we find is that the dentate and CA3 in young adults are relatively high the entire time. 0, by the way, is novel foils, so it's brand new items. So that's actually the highest point that you can get to. And what you see here is that in older adults, there's a much more linear kind of pattern here, consistent with the idea that they need things to be different sufficiently different in order to call them different. Okay? So now this is great. So we have evidence for reduced discrimination. There's a neurobiological substrate. There's a part of the brain that seems to change in a way that reflects this behavioral change. And the question is, why does this happen? And one answer to this, and there may be many answers, is that there's hyperactivity in one of these regions that drives it towards generalization. And there's a pretty complex story behind that, so I'm not going to go through it in detail. But suffice it to say that there is evidence for this hyperactivity, and it does predict the extent to which animals are impaired on these kinds of tasks. So that's what you see here. This is during new items or familiar items. You see a higher firing rate in the rats, but we also see the same thing in humans. In fMRI signals, we've been able to document hyperactivity also during encoding and retrieval, and the extent of this hyperactivity predicted how much of a discrimination loss they had in their behavior, which I think was a very necessary thing to show in order to say that this hyperactivity is bad. It's not a good thing to have this hyperactivity. So now one question one can ask is, what about reducing this hyperactivity? Can that be done? And what would that do to memory? Can it actually help rescue some of the memory deficits? So we ran a proof of concept clinical trial a few years back. This is in healthy controls at N of 17 and AMCI, amnestic MCI patients. This was a randomized double-blind placebo-controlled trial, so it was a placebo control that we used, and we used the drug levetiracetam, which is a very well-tolerated, FDA-approved, known in the literature for quite a long time, anti-epileptic. So what we've been able to do here is, using this low dose of levetiracetam, be able to bring down the hyperactivity in the dentate CA3 in the left-treated participants. Also, we found that in AMCI there was a hypoactivity. There's a loss of activity in the lateral entorhinal cortex, one of the input routes into the hippocampus, which I'll come back to a little bit later. But that activity also was rescued, so it was brought back up. Right? So not everything diminished, some things came back up, and behavior was actually improved significantly also in terms of discrimination. So this was very exciting for us, and we started to think about ways to be able to talk about this a little bit more outside of the context of, well, look, this is an anti-epileptic, so it has to be about epilepsy and Alzheimer's disease. Keep in mind, these are not epileptic patients. And this is not a dose that is effective for epilepsy. This is a dose that is an order of magnitude lower than you would give to an epileptic patient. So we're talking about a very different kind of entity here, but this is sort of a repurposing of that already approved drug. Now, the question is, why is hyperactivity bad? So we've determined that it is bad, at least in our case, because it correlates with impaired behavior, but why? What does hyperactivity actually do to synapses? And one hypothesis is that it actually might drive a stress response and might cause dendrites to retract in the hippocampus. It might actually cause a synaptic disconnection in the hippocampus. So for example, the perforant path, which is a pathway that leads into the hippocampus from the rest of the brain, there's pretty good evidence that dates back as early as in the late 70s. There's pretty good evidence, at least in rodent models, that this perforant path degrades as a function of age, and much more severely so in Alzheimer's disease. So this is just sort of reminders of these data from way back when, but what we've tried to do is actually try to look at this in humans. So now I'm shifting to another modality, not fMRI. Now we're going to talk about DTI, or diffusion tensor imaging. Now, diffusion tensor imaging is a method that allows us to map water movement, and water is always in constant flow in the brain, but wherever you have structure that constrains its movement, like the axons, for example, the fibers, the tracts, that kind of structure constrains this movement and makes it much more directional. And directionality of that signal is what we can measure in this imaging technique. And this is the kind of tractography images that one can find from this. But what we've been able to do is actually increase the resolution of this diffusion tensor imaging technique such that we can look at something like the perforant path. So this is just for the methods weenies or the enthusiasts. I'm going to leave it up there just for a few seconds, and if you want to talk about it, we can talk about it later. These are just details of the protocol that we used. But ultimately, I want to show you what the data actually showed. So this is where the perforant path is supposed to be based on retrograde tracer studies, and this is one of the images from our study here, and you can see that this is the directionality of it. It goes from the entorhinal cortex into the hippocampus. We can quantify this signal. We can actually do tractography with it. So this is not one of ours. This is actually from a colleague, Jean Augustinac at MGH, who is an anatomist and has done this with postmortem data. And you can see beautiful entorhinal cortex here and hippocampus here, and this is the performant path connecting them. I wanted to show you this because this is sort of validation for our approach, where we've been able to find signals that are very much similar to it. Of course, our resolution is nowhere near as high because these brains were dead, so you can keep them in the scanner for quite a while, whereas our subjects were very much alive and we wanted to keep them in there for less than 45 minutes. So we can do tractography with it. We can also quantify its integrity, and we came up with a method to do that. We basically just draw a line across the back of the entorhinal cortex, and we ask how much of the diffusion signal is going directly perpendicular to it, which is the directionality of the perforant path. We can quantify this across different voxels. This is sort of methodsy stuff you don't need to worry about so much, but we can essentially quantify the integrity of this function and be able to compare it on a subject-specific level and look at young adults versus older adults versus MCI and so on. And what we've been able to find is that calculating, for example, a measure like area under the curve, we can compare young versus older adults, and we see that there's a clear age-related degradation of the perforant path. And it's specific. It doesn't happen in other hippocampal pathways. For example, the alveus, which is another sort of control pathway that we used here, didn't have that. Now, one issue is that some of these subjects may actually have had Alzheimer's pathology. We don't know because we didn't do CSF on them, we didn't do amyloid imaging on them. That's the next step that we're working towards now. So it could be sort of intermixed healthy older aging with pathological aging. We do know that that feature is linked to their memory performance, so the integrity of their perforant path was directly correlated with their discrimination performance and also with their performance on the Ray Auditory Verbal Learning Test, which is a word list learning kind of test. So that gives us a little bit of external validity as well. Now, the key for this perforant path is that it connects, again, the entorhinal cortex to the hippocampus. And the entorhinal cortex, you don't hear it too much in Alzheimer's conferences. You always hear about the hippocampus, but the entorhinal cortex is very important because that is one of the very first places where you start to get tangle pathology. And it's one of the first places where you see evidence of neurodegeneration. So we've focused also with a laser on entorhinal cortex. Now I'm switching to talk about structural MRI data, and we've tried to measure its thickness and see if actually that predicts whether or not somebody is declining or not. So here's, this is data from the ADNI study, the Alzheimer's Disease Neuroimaging Initiative, and we can see that there are clear thickness changes in AMCI and much more so in Alzheimer's disease, but there is no evidence of such a decline just in healthy aging. So as you look at the entire healthy aging sample here, it's pretty much a flat line. Okay, so this is really an AD-specific kind of biomarker. Decline over a 1-year period happens in an MCI group, does not happen in the healthy control group. You can see that the slope of decline can actually distinguish those two groups. And it's interesting because this change is detectable even at periods as low as 6 months. So this is a pretty sensitive kind of marker. We also see that it's linked to cognitive outcomes or clinical cognitive outcomes because we can take a look at people's CDR sum of boxes scores and ask the question, if you have a CDR sum of boxes of 0, for example, or 0.5, when you decline on that CDR sum of boxes, was there something at baseline that allowed us to predict that decline? So we took two groups. One is the CDR sum of boxes that was stable over a year and one that was declining. And what we see is the entorhinal cortical thickness was actually predictive of the slopes of, or the slope of the entorhinal cortical thickness was predictive of whether or not people were stable or declining. And that you can see here. It's a 1-year change in EC thickness. We also can ask the question, what about amyloid pathology? So, for example, we can ask the question of if somebody's amyloid positive versus amyloid negative, do we think EC thickness is going to decline much more rapidly in one versus the other? And we found something very interesting there. So this is looking at CSF A beta using the published cutoffs on this. We find that at baseline it wasn't so much distinguishable, but the slope was certainly distinguishable. This is in AMCI patients. So slope over 1 year certainly distinguished the two groups from each other. But then we asked, in my opinion, the more important question, what if we don't look at AMCI? What if we look at healthy controls? And ask the question, if you're amyloid positive, are you going to be more vulnerable to this kind of neurodegeneration? And the answer was sort of. So even though there is a change in the slope, but the larger change actually was at baseline. So even at baseline, these are all healthy asymptomatic older adults, right? But some of them are amyloid positive and some are amyloid negative. These are the negative ones, these are the positive ones. And what you can see is that the slope actually is different between those two, and at baseline, they are different. So there is good evidence to suggest that there is sort of a combination of things happening in this medial temporal lobe, entorhinal hippocampal kind of network. Some are functional, some are structural, some are based on diffusion. but it's a combination of different neuroimaging biomarkers. I should also mention we've done some control analyses, like primary visual cortex, for example, doesn't change, and we certainly don't see any change either in normal healthy controls or AMCI. So this is something that is a little bit more specific. But there's something else that happened recently that was very exciting, which is a work that was published by colleagues at Columbia. This is from Scott Small's group, looking at lateral entorhinal cortex and the pathology therein, and proposing that through a prion-like mechanism, pathology might actually spread from the lateral entorhinal to other sites. And we have now a way to be able to measure the thickness, particularly of the lateral entorhinal cortex, although I'm not sharing that data with you today. But this is the kind of thing that, so I wrote a preview for them, and this is the kind of thing that we are now also proposing, which is that the pathology starts in LEC and it really spreads to other sites, and we think we have a mechanism as to how it manifests early on here. Now, in terms of hypothesized mechanisms, this is kind of the summary. Let me just walk through what we've covered here. This is a schematized representation of hippocampal connectivity. These are the two regions where we saw hyperactivity in the past. This is the lateral entorhinal. PP stands for perforant path, and so on. So the idea is that we have hyperactivity that manifests early on in CA3 because of synaptic dysfunction. There's a bidirectional relationship between amyloid and hyperactivity. So this is an evolving story, but it seems like hyperactivity can actually induce amyloid plaque deposits. And wherever you have clusters of plaque deposits, you can also have hyperactivity that is induced. So there's a bidirectional link. Hippocampal stress in this network seems to retract the apical dendrites in CA3, which make contact with this perforant path, which ultimately will result in the elimination of this pathway. And this happens in the context of just older adulthood, but it happens much more dramatically so in AMCI and Alzheimer's disease. And once that's reduced, you start to get some of the symptoms or some of the changes in the lateral entorhinal cortex here, and the first way that we can pick it up is through neurodegeneration. Obviously, this is also where tangle pathology is forming, but we can only pick that up so far with autopsy, maybe soon with an imaging technique as well. But the idea is that from there, it will really spread to other sites, some of which are neighboring and some of which are farther apart. But these are really all of the players that we've known about for quite some time. These are the kinds of things that come up when we talk about the networks involved in Alzheimer's disease. So this is the kind of story that we've been thinking about, and the kinds of things that we've been trying to gather evidence for. One of the things that has been really inspiring is the model by Cliff Jack, which allows us to sort of look at the temporal progression of these biomarkers. But I do have to say I disagree on one thing, which is that MRI and FDG PET, all MRI and FDG PET is kind of clumped together into one line, and that's what you see here. And I would argue that MRI is not a one-size-fits-all kind of phenomenon. There's actually quite a bit of change and differences in terms of the types of modalities that one can use. So functional things will happen first. Those are correlated to synapses. Structural things will happen later on, and we have techniques to be able to look at all of these things. So my version of this, for example, to be able to add a couple of things onto it, is that there is hyperactivity that manifests pretty early on, and we think that's related to amyloid. There's perforant path pathology that builds up sort of middle of the way before entorhinal cortical degeneration, and entorhinal cortical degeneration will happen at the same time as you're building up tau deposits. So there are at least 3 different families of biomarkers that can be looked at that really vary across that temporal spectrum. One critical thing to think about is really what the space is going to be. So if you're trying to look at things in preclinical AD, the kinds of biomarkers and how dynamic they're going to be is going to be very different from looking at symptomatic phase or MCI. So some of these biomarkers will be much more dynamic here, some of them will be much more dynamic here. Certainly by the time you get out to AD, very little is going to be dynamic at that point aside from just structural degeneration. So it's important to keep this in mind, and we're trying to sort of fit squarely in this space here of preclinical Alzheimer's disease, because I think we'll all agree that the earlier that we can target the disease, the better, and we're trying to use this also to enrich clinical trials with the right kinds of biomarkers. So just to circle back to the take-home messages here, memory changes in subtle ways as we get older. We all know about this, and I've talked to you about the kinds of cognitive complaints that people have talked about for many, many years, but it's important to think that the discrimination kind of change that happens is really one that spans across many of these memory problems. And I showed you what happens when we have an experiment or a task that is targeted directly to that. Now, high-resolution imaging techniques, I showed you 3 different flavors. There's structural MRI, there's functional MRI, and there's diffusion MRI. And each one of these gives you a very unique picture, very different idea of what's happening. And some of them are able to tap into neurobiological substrates that are more related to synaptic dysfunction, sort of, the connectivity kinds of changes. And some are much more on the structural side, so they're going to tell you about neurodegeneration, cell loss. And it's not clear yet if these things are exactly as clear as, so I make them sound very clear, but it's important to remember that we still need validation of all of these things, right? So in order to say that structural MRI is going to index cell loss, I need to be able to do structural MRI in an animal and then be able to sacrifice that animal later on and determine that it was truly cell loss and not something else that's driving it. And this is one of the reasons why my work has been here at UCI MIND, because we have these wonderful animal models, whether it's mice or rats or any other model. These kinds of things allow us to test these hypotheses with a fair degree of precision that we can't do just by looking at human data. Identifying these changes, I hope I've made the case that they're important for understanding the neurobiological substrates. I've shown you one example of what can happen if you pick up on a new feature, so kind of like this hyperactivity, and try to target it in a clinical trial and actually get rescue. So this is it was a phase 2 trial, so it's not clear whether or not this is going to also translate to a phase 3 trial. That's what we're trying to get the funding for now, to see if it'll work in the long run. But those are the kinds of things that imaging biomarkers can give you that are very, very different from the targets or the usual suspects like amyloid and tau. These brain imaging biomarkers, again, can be used as indicators in the absence of symptoms. You can also use them when you're talking about MCI and Alzheimer's disease, but I think the best kind of use is going to be really in the preclinical realm. And this talk is really focusing on just using it for research tools, but there's another talk by Liana Postolova. I think you guys, by being here, you probably missed it, which is talking about the clinical kind of use or the diagnostic value of imaging. And it does have a lot of diagnostic value as well. And finally, I think that a paradigm shift to talk about mechanistic biomarkers that can be derived from neuroimaging substrates is really, really important. And it's not easy, and all of these neuroimaging biomarkers don't have very, very straightforward kinds of explanations, but neither does any of the biomarkers that we have, I would argue. But I think this is a very important step to be able to move forward on this. So finally, I want to acknowledge the people who actually do all of this work and allow me to take the credit for it. This is actually a picture of my lab from back at Johns Hopkins. We only moved here very recently. These are the folks who contributed at JHU. Many of them have come with me on this trip to UC Irvine. And these are our collaborators and the support that we get. And I thank them all for their contributions, and certainly thank our supporters for many, many years of support for this work. And thank you guys for listening.